dugsiga boardingka iyo maalinlaha ah ee Amano Boarding and Day School.
Everybody, and I welcome you to today's tutorial. In our last tutorial, we were looking at coordination. We looked at coordination and homeostasis. Now today, I want us to look at homeostasis and exclusion. And when we talk about homeostasis, I want us to first look at urine, because it's one of the waste materials that are always removed from the body. For a normal human being, every day produces around 1.5 liters of urine or 1.5 decimeters of urine. But the volume of urine produced depends on the amount of water you take in and the amount of water you lose through the mechanisms we looked at last time. Like we said, we lose water when we sweat and also through feces. But out of every liter of urine, at least 40 grams is for waste products. Then the rest, water. Then we come to the composition of urine. It's made up of mainly urea, and you can see urea is carrying the biggest percentage or amount. Ammonia. We also have other nitrogenous wastes. We say others because we have talked about urea and ammonia. They are nitrogenous. They contain nitrogen, but there are also others. Salts like sodium chloride, potassium, and phosphate. Then when we say nitrogenous wastes, we mean waste materials that contain nitrogen. And when we talk about exclusion, simply we mean removal of nitrogenous waste products of metabolism. It's not removal of waste product, not all waste products, only waste products of metabolism. And when we talk about metabolism, we mean biochemical reactions or chemical reactions that take place in cells of living organisms. So we have egestion. Egestion simply means removal of undigested food in form of feces through the anus. So that is not exclusion. The reason why it is not exclusion is because the feces or the undigested food contains very few or no waste products of metabolism. It doesn't contain any waste product that contains nitrogen. Therefore, we don't consider it as exclusion. And another reason why we don't consider it as exclusion is because feces, they don't cross any membrane. From the time you take in food through the mouth, that food will move down until it moves out without having crossed any membrane. But these other nitrogenous wastes, already they are produced inside the cell, so they move out of the cell by crossing that membrane, the cell membrane. But these ones, they don't cross any membrane, and therefore they don't qualify to be called nitrogenous wastes. So we have this structure of the urinary system. It has these organs. It has two kidneys, as you can see. We have the left and the right. Then and also it has this urinary bladder, which temporarily stores the urine, and then the urine is brought to the urinary bladder by these two ureters. We have the left and the right. The left carries from the left kidney, and then the right carries from the right kidney. Then we have these two blood vessels. We have the renal artery and the renal vein. The renal artery is bringing in blood that is oxygenated and full of waste material. And then the renal vein is returning the oxygenated blood that is free from waste material, because already it has been filtered in the kidney. Components of the urinary system, we have already seen many of them. So I've talked about the urinary bladder, which temporarily stores urine. I say it is temporarily because once it gets full, urine has to be removed. The two ureters, these transport urine from the kidney to the urinary bladder for temporary storage. We have the urethra, which is an opening from the urinary bladder through which urine passes to outside. And then when you look at the urethra, being an opening from the bladder, it has two rings of muscles we call sphincter muscles. We have the lower one and the upper one. But the lower one is always under conscious or voluntary control, meaning that its contraction is controlled by the brain. You decide. You can make it contract or relax and pass out urine. But the upper one is under involuntary control. Its contraction and relaxation occurs without awareness of the brain. That's why sometimes when the bladder is full, you lose that control and urine can get out. Then the urinary artery and the urinary vein, I've already talked about them. We said the urinary artery brings in blood and it branches directly from the aorta. Then the renal vein, it returns cleaned blood or filtered blood from the kidney to the main vein, which is the vena cava. Then when you look at the kidney is the main excretory organ. And there too we have the left and the right. So we have this structure of the kidney. As you can see, it is bean shaped. It has this outer layer, the dark layer, which we call the cortex. And this cortex has many branches of renal arteries. They are branching from the renal artery and many nephrons. Then next to the cortex, we have another layer we call the medulla, which connects to the pelvis. And then this nephron, once reabsorption is done, the waste material or the urea moves up to the pelvis, and then the pelvis connects to the ureter, which carries the urine to the urinary bladder for temporary storage. As we've already seen, I said the outer layer is the cortex with many branches of arteries, and then we say that the pelvis joins the ureters that carry urine to the urinary bladder. Then when you look at, already we say that in the cortex and the medulla we have those nephrons. A nephron is the smallest and basic functioning unit of the kidney. And when you look at each kidney has millions of nephrons. So here we have the structure of the nephron. We have this part we call the glomerulus, where we have the blood, the artery bringing in, or the efferent bringing in blood with waste material. Then filtration happens here. Then this blood returns to the heart free from waste material. Then it has this coiling, which we call the proximal convoluted tubule. That's the first coiling here. Then we have this U-shaped tube, which we call the loop of Henry. And then this second coiling is what we call the distal convoluted tubule, which connects to the collecting ducts. These are collecting ducts. We come to ultrafiltration. When we talk about ultrafiltration, we mean the process by which small-sized molecules or materials pass through the walls of the granulars into the Bowman's capsule to form a granular filtrate. Then, a blood vessel, when you look at the Bowman's capsule or the granulars, a blood vessel that brings in blood has a bigger diameter, while the blood vessel that carries blood away from the granulars has a smaller diameter. So meaning that more blood is coming into the granulars, but less blood is leaving the granulars. So as a result, pressure builds up in the granulars. And then this pressure forces the small-sized molecules to pass through the walls of the granulars into the Bowman's capsule. And that process is what we call ultrafiltration. When you look here, you can see that as pressure builds up, these are the walls of the granulars. So small-sized materials pass through and then go up to the proximal converted tubule. They begin to move along the nephron or the tubule. And then as reabsorption takes place, by the time this granular filtrate reaches the collecting ducts, all the essential materials have been reabsorbed, and then the rest is lost as urine. So this is an extension of the granulars. This is the membrane. You can see it has pores which can allow small-sized materials to pass through. So thank you for watching. Have a good day, and please try the assignment that is attached together with this video. Thank you.